Okay, good morning. Thank you guys for taking time to come in. This is a job candidate coming in for the University of Alabama. And so the talk title today is Building Coral Resilience in a Brave New World. The guest speaker today is Raphael Ritson-Williams. Rafi did his undergraduate at Evergreen State College in Washington. He then went to the University of Guam and worked with Val Paul. And then he stayed on in Val's lab for several years after that as a tech and a research scientist and a whole bunch of different kinds of roles for about 10 years and published a whole bunch of really great papers in coral and algae. And then he went to the University of Hawaii and did a PhD with Ruth Gates, doing some really cool genomic work on that system. And finally, he's now a postdoc at the California Academy of Sciences. So help me welcome Rafi, and we look forward to your talk. That's a clap, guys. There you go.
Thank you so much, Lee. Okay, good, this is on. I'll try not to speak too loud. I tend to be loud and obnoxious, so whenever you give me a microphone, it's even worse. But thank you all so much for coming. I should say early on, vote for me, because I'm a candidate, and you'll see more candidates. Into the science. So hopefully you guys seen this title, this Brave New World, and that sort of makes you think of the literature arts and this dystopian future. And I think while the focus of this talk is really about corals, most of my research has been on corals, clearly these techniques and concepts could apply to any organism, and I would never say I limit myself to only corals. They've become a poster child for climate change, and so they're sexy and fundable. But there's a lot of similar questions in different systems that we can ask. And you can see there's a nice framework from this stuff that I'll present. And so one of the critical things we're sort of approaching as climate change and other human local impacts are happening more and more frequently is what builds resilience? What are the components of resilience, and how can we help or damage that type of resilience? Clearly, we can't manage resilience if we don't understand what the components are. Resilience isn't a single thing. It's actually this interaction of two things. Both the ability of a system to resist stress events and the ability of the system to recover from those stress events. So think of a rubber band. You poke the rubber band and it moves back, and then it snaps back, right? And that's really important. People throw this term resilience around a lot. So I think it's important to define it. Critically, it's both resistance and recovery, so I'll cover both of these types of processes in this talk, even though they're from sort of two disparate types of experimental systems. I like to show this picture because this is a lava flow on the Big Island of Hawaii that happened in 1970, and clearly there's no way an organism can resist hot molten lava, right? You can't stop. You can't save yourself from boiling Earth, right? But there is some resistance in this habitat in terms of up the slopes of the mountain. The native forest still persisted, and having this refuge is really important for resistance, and that's some of what I'll talk about. This bay I worked in was sort of a refuge from climate change. And then recovery, and so this is an ohia tree. For those of you who have never been to Hawaii, ohia is sort of a foundational species of their forests, and it's one of the most important species because all the native birds feed on these flowers. And what you can see in this picture that I think is really special is this is a really small ohia. It's about up to my waist, but it's already flowering. And so this is a sign that it's reproductively mature, and it's already recolonizing this habitat. And that recolonization is really critical for this idea of resilience, right? So this is just a great example of sort of the types of topics you could cover in any system. Again, I'm interested in coral reefs. A lot of what we're seeing on reefs is that reefs are driving from coral-dominated states to macroalgal-dominated states. I put this question mark here because I would really like to know the research that drives them back. I really want to answer these questions. What are the processes of restoring reefs to their coral dominance so that they have their sort of natural functions? I did my PhD in Hawaii. which was terrific. One of the great advantages was that I was working on a coral reef, and so I could go out and look at a coral reef all the time. When I started, I had this big idea, and I had written down a big proposal, and everybody was like, okay, you should do this experiment. And then that summer there was a big bleaching event, and I was like, okay, throw that out the window. I'm going to go out and study bleaching, right? So we would see things like this in the field. Pretty huge parts of the reef were bleached and stressed out. And so I was very interested in this sort of stress events. And so if you don't know, coral bleaching is sort of breakdown of symbiosis. And this symbiosis is really critical for corals because it's how they get a lot of their nutrition. And so a coral is an animal, and the animal hosts Symbiodinium, which is a single-cell dinoflagellate. That Symbiodinium photosynthesizes. And so I compare this to paying rent. And so the coral animal gives the Symbiodinium a house to live in, and the Symbiodinium provides sugars as rent. They're paying rent to live in this place. And that symbiosis has existed for millions of years. It's what's allowed corals to be so successful. A lot of their calcification is driven by the sugars that are produced by this photosynthesis. So when seawater temperatures get too high or other types of stress events happen, this symbiosis breaks down. We still don't really understand, but we might be getting at that, and I'll show you some of that data. We don't really understand who's in charge of this breakdown. We know that this photosystem 2 stops working and that the symbionts produce reactive oxygen species, and this is toxic to both the corals and the Symbiodinium. And so either the Symbiodinium leave the cells because the landlord didn't provide the right air conditioning, or the coral kicks them out because the Symbiodinium is no longer paying rent, right? And so we don't really know which one's in control. But clearly, bleaching is this process of a breakdown in symbiosis. And that doesn't mean the coral is dead. If you can see, if you can look really closely at this picture, I know it's far away, there's a little polyp right there. That coral animal is still alive. This is a stress response. What we're seeing is stress for this creature, but it can recover from that bleaching, and it can sort of repopulate the Symbiodinium over time, assuming that the stress that caused the bleaching disappears. So I've always been sort of a positive person, and I'm always looking for bright spots, and I'm really curious about not what's killing corals, but why some corals survive these stress events. And I saw a lot of this in the field, and this was really interesting. This species, Porites compressa, was fully bleached. Right next to it, another colony of Porites compressa hosts the same symbionts, is completely resistant to this stress. Clearly, these are in the same microhabitat, so they're experiencing the same seawater temperatures, but there's something special about this colony that's allowing it to resist stress. What is it? What's the components of that stress resistance? That's sort of the foundation of my dissertation. And so I was really interested in this question. We can chase this in all sorts of ways. I was lucky to work in Kaneohe Bay. This is the Hawaii Institute of Marine Biology. It's a teeny island in the middle of this huge bay, right? It's beautiful, gorgeous place to work. You can't get to the island except by boat, and so you park in this lot, and then your morning commute is a little boat ride across the island. Much like if you guys didn't have a causeway, right? You'd have to take a boat to work, which could be fun, but it really sucks when it's raining. So I wouldn't recommend getting rid of the causeway. But what's exciting about working at a marine lab like this is if you're studying corals, there's corals surrounding this island. All through this bay, there's a bunch of little patch reefs that make individual little replicates, and then they're across an environmental gradient that's produced by the bay with multiple inflows and all sorts of sort of physical parameters that you can study. So in both 2014 and 2015, there was a bleaching event, and this was mostly driven by an elevated seawater temperature. So the normal maxima seawater temperature in Hawaii is about 28 degrees, and you can see in both years we went past 28 degrees. Once you go past 28 degrees, corals start to bleach. Once you go past 30 degrees, corals start to die. So we were sort of intermediate in that type of stress, and you can see in 2014 we had a different type of thermal stress. Around August we had a sort of a peak, and corals started to bleach. They were really maximally bleached in September, that maximal time, and then we had kind of a storm event, really windy weather and some rain that really decreased the seawater temperatures very rapidly. And so a lot of the corals I studied recovered. Again, in 2015, we had a thermal stress. This one started a little earlier. The temperatures got pretty high in June, July, and then we started seeing bleaching, and then the bleaching persisted longer this year. It went across the whole summer to September. And then again in October, we were lucky we had a storm event, and that really cooled down the seawater where these corals were exposed to this thermal stress. This is a map of Kaneohe Bay, and so you can see every number is a different patch reef. HIMB is the marine lab where I worked. Reef 25 is sort of an intermediate reef. It's in the middle of the bay. It doesn't have a lot of freshwater input. And Reef 44 is right at the mouth of a river. This would be a river if you could see the rest of this thing. And so I monitored and collected data from all three of these reefs because I was really interested in sort of environmental variation across the bay. I really like to correlate sort of abiotic factors with biologic response. It's critical in ecology to understand those types of things, those types of interactions. So I spent a lot of time monitoring temperature at each of these reefs. We didn't see any difference in temperature. Monitored light at each of these reefs. We didn't really see any significant variation in light across these sites. And I monitored other physical parameters, nutrients and sedimentation. Not a lot of differences. So we're calling the bay sort of a very similar system for this stress response. So even though I set up reefs at multiple sites, those you can think of those as kind of replicate sites that are experienced effectively the same abiotic conditions. And so I was really interested in what the future is going to happen on reefs, how climate change is going to change diversity of reefs. Kaneohe Bay is really dominated by two species of coral, this Porites compressa, Montipora capitata. So when you run a video transect, which I did a lot of, you're really sort of counting one or two species. You're missing some of the smaller members of the community, and I was really interested in how the community was going to shift in the future. So I took the time to do these timed swims. So I swam around the reef for 30 minutes, and I counted every coral colony of every species that I could find, and I categorized it as bleached or unbleached. The white part of these pie charts is bleached, so 90% of Poecilopeira damocornis bleached in 2014, and the black part of the pie chart are relatively healthy colonies. And you can see it could do that for 2 years, and you can sort of get this sense of whether this species is very susceptible, which is Poecilopeira, to thermal stress, and then one of the dominant species, is a little more resistant. Porites is even more resistant. And this little guy, Leptastria purpurea, is really amazing. It's tiny, right? It's this little sort of button-sized coral. But something really special about this species is it completely resists bleaching. I must have counted thousands of these little individuals, and only one of them the whole time, both years, ever bleached. So I'm not going to talk anything about this species, but this is a really cool follow-up. I think for my dissertation, there could be a lot of really interesting things about what's different among species in bleaching susceptibility. But regardless, we get sort of an index when we do this kind of stuff, and so you can start doing sort of higher-level statistics, a two-way ANOVA, and see if there's difference in susceptibility among species and then among years. And so you get something like this. Dotted lines are significant groups. We see that there's certainly Poecilopeira, these top two species, are very susceptible to bleaching, whereas there's intermediate species that aren't quite as susceptible, and Leptastria purpurea is really resistant. And so this is sort of a prediction of what the reefs are going to look like in the future. We can sort of start saying that Poeciloperas are going to die. Those are going to disappear. And then there's going to be some variable responses, and the reefs of the future are going to be composed of these teeny little button-sized corals. So maybe not so useful for ecosystem services. So that's important. One of the things that was most interesting to me was that there was a difference between 2014 and 2015. There was much less bleaching in 2015. A lot of my colleagues were like, well, this is clearly a sign of acclimation, right? We know that corals are bleaching less in the second year. And I was like, well, this isn't actually the right data to show that. We don't know. Maybe there were differential mortality, and so there just weren't as many coral colonies to measure the next year. And so then it looks like there's less bleaching because there's less things on the reef. So a much better way of getting to understand that is actually to look at individual coral colonies and see if they had the ability to resist bleaching over multiple years after 1 year of bleaching or not, right? And so to get at that question, I went out and I tagged a bunch of corals so I could monitor the same individual corals over time and space at all three of these reefs. And I tagged them very specifically as pairs because I was really interested in comparing these types of phenotypes, right? So this phenotypic variation is really important. It's fundamentally the the sauce of natural variation that we would expect natural selection to act on, right? And so we're actually sort of getting to the components of evolution when we look at this phenotypic variation. But what's driving that variation is sort of this fundamental question that nobody really knows the answer to in corals. And so I tagged a bunch of these. Over 200 corals were tagged as these pairs. And then over time, because I was in this really great situation of being in Kaneohe Bay, I could go out and check those same corals every month. I could take samples of them over months. I monitored these for 3 years. Here's a sample of one pair, 51 and 52 from Reef 25, that I started looking at in October. They were fully bleached, and then they started to recover. And you could see that their tissue is darkening a little bit, right? Of the bleached one, the whole time 52 was fine, right? It didn't bleach at all. And then what was sort of fortuitous about this is it allowed me to look at them the next year. And even though we didn't know bleaching was going to happen in 2015, it started to happen again, starting in July. August, you can see the colony's getting quite white. September, it's fully white. It's fully bleached again. October, it's still pretty stressed. And then November, it's starting to recover a little bit. And it's coming back dark again in December. What was amazing about these two phenotypes were they were consistent among the two thermal stresses, right? And so that's really exciting. It also means there's no acclimation going on. It means those colonies that bleached again the next year. And so I think in terms of whether we saw in situ acclimation, we really didn't see that very much in this system. But it shows that the phenotypes are consistent. It wasn't a one-off. It wasn't just this random response to this one type of thermal stress. In both of those thermal stresses, we had the same two phenotypes. We did acute quantitative PCR to measure the abundance and the type of Symbiodinium that were in these corals. And Montipora can host both C and D clades. We know D is thermally resistant, but C we know a lot less about. And these are the three different reefs. This is sort of the abundance of zooxanthellae. So any dots that are lower are bleached, and then the top line are all those colonies that resisted bleaching, right? And you can see over the 2 years we get really similar patterns. We have sort of this recovery rate, we have a healthy time period, and then we have a bleaching and recovery rate. And this was sort of really cool. We didn't see very many signs. Oh, whoops. That slide is gone. We didn't see very many signs of shuffling. So there's this idea in the literature that if you host clade D, you would be resistant to coral bleaching because clade D is thermally tolerant, but it doesn't give you as much carbon as C does. And so we measured the abundance of C and D. If a coral hosted clade C, it kept clade C even after bleaching. And that was especially true in the first year. In the second year, we found 3 colonies out of 50 that did shuffle. And so we think it's possible that these corals can sort of adjust their physiology to having new symbionts, but it's quite rare. And we don't think it's probably as common as you might expect from reading it in the literature. So we can sort of get at this question that, is it actually the C or D interaction that's important for resistance? And what we see in the bleached corals, anything with B is bleached. Clearly, there's less abundance of zooxanthellae. We know that. That's the definition of bleaching. But in the unbleached corals, we had both C and D. And this was really interesting because we sort of assumed D was going to be the major component of the unbleached corals. But actually, there's quite a few C. These are the same C types as we see in the bleached corals. So there's something else going on here other than the type of symbionts it's hosting. Oh, sorry, there's the slide. Where we see pretty low levels of shuffling. So for the most part, these top bars are those colonies that did not bleach at all, and you can see as you'd expect, they maintain the same symbionts. There's a lot of sort of physiologic regulation to interacting with these symbionts, so it makes sense to be consistent. But in thermal stress, even these Cs never changed. But in this bottom figure, we have something like 7 colonies that actually did shuffle. Even some of the ones that didn't bleach shuffled, and this makes a lot of sense to me in terms of they had D during the bleaching event, and then they had C when it didn't bleach, and that's really like optimal for these corals because C is producing way more carbon for the coral host, and so it's a much better renter. And then we have some interesting things here with that same sort of trajectory where the bleached corals had C, and then after this, even though there was a little bit of D after the first bleaching, very little, and that all went right back to C But then after the second bleaching, there was 3 colonies that shuffled to D. So that's an interesting strategy. We think that maybe multiple stressors is impacting that. It's also 2 different types of thermal stress. And so we don't know if it's the extent, the duration, or the maxima, thermal maxima that's driving this. These are all excellent experimental questions. We can get it in the system because we have tagged these corals. So we know the genotypes that shuffle and the ones that don't, and so we can always go back to these corals and ask these types of really interesting sort of physiologic questions, right? But Porites compressa also had these 2 phenotypes in the field, and we know it's almost one type of Symbiodinium, C15. So I was really interested in the sort of genetic architecture, and so what does any of the genes inside these corals, is that what's allowing them to resist bleaching? And that's just sort of measuring sort of genetic diversity among sort of classic FST, measuring diversity among populations, right? So I did this RAD-seq method where you take the genome, use enzymes, chop it up into a bunch of little bits, and then you can assemble these enzymes into these stacks, these genes into these stacks. And these stacks, you can imagine every line could be an individual coral, and every individual gives you the same gene region. It's usually 100 base pairs or 150 base pair long to compare. And then if you have differences among the population that correlate with your environmental stress, you would see something like this. Half the population have a SNP of C, and the other half have a SNP of A. And so this is a pretty powerful way of randomly sampling genome when you're a grad student and you don't have enough money to do whole genome sequencing, right? And so this was kind of a nice technique that I sort of threw at the system, and we do all the analysis in the bioinformatics, and we get this sort of result where we see outlier SNPs. So there's at least 160 sites that varied, these loci that varied between some of the corals, and there were 2 groups, and I was really excited, and then I made this PCA plot, and the groups have nothing to do with bleaching, right? Classic sort of grad student project. And so what we saw was that there was sort of a mix in these 2 groups of bleaching and unbleached, so that was sort of confounding. So I threw another factor at it. We had them collected them from 2 different sites. Maybe they were reproductively isolated somehow in the same bay. It doesn't look like that's happening either. It's not a distance question. So I annotated there's about 100 genes that make up these 2 groups, and I annotated a bunch of those. I could annotate about a third of them, and we found quite a few of them were mitochondrial genes. And these are classic species level markers, and so it made us wonder if Porites compressa has multiple species. So I got together with some of my collaborators at HIMB. Zach Forsman had a bunch of unpublished data for Porites lobata, and we started comparing among species of Porites. And so Porites evermani is a massive species that seems to be a true, good individual species. Porites compressa is this branching form. Porites lobata is a massive form. Clearly 2 different species, right? And then when you do the genetics, you start seeing trends like this. And so this blue dot is all compressa. These yellow dots are all lobata, and then we have this really interesting clade in between the 2. And we think there's hybridization going on. These lineages aren't reproductively isolated, and so there's some way that they're recombining with each other. With these 2 groups that I found were actually sort of a signal of multiple clades and evolutionary trajectories going on in these species. So, you can't trust morphology, right? Don't judge a coral by its cover. So we're kind of in that situation. And so this is really exciting data in terms of like what is a species. Corals have always challenged the notion of species. If you want to get in a really long conversation with me, come and ask me what a species is later, and we can discuss that. But at least from this analysis, and I can't rule out that there's not genetic architecture that does support these 2 phenotypes. We just did a random sampling of the genome. The next step for this project is to do whole genome sampling, sequencing, right, and actually get at sort of the whole genome at low coverage, compare among the individuals, and look for phenotype differences that might be there. But that's really expensive, and so that's sort of an NSF sort of style grant. But we moved on to another technique. We wanted to know if the microbiome were influencing this, not just the Symbiodinium dinoflagellates, but if the bacteria that live within corals, right? Corals are this really holobiont type of thing. It's a lot of organisms all living together under the hood of this coral animal. So we did 16S amplicon sequencing, and 16S we kind of use as a barcode for microbe species, whether it's a true barcode or not, we could argue about that too. But it's sort of the state of the art right now. So we get these sort of community patterns, and we did see quite a few differences. It was kind of interesting. So in this figure, this is relative abundance of the microbial creature, and in this case, we're looking at cyanobacteria. We've lumped a bunch of OTs together under the sort of general term cyanobacteria. And the red lines are the bleached corals. Bleaching happened in October, and again in September. And then during November and January, there's recovery going on, and we think of May as sort of fully recovered. They were fully dark. They had regained their whole population of Symbiodinium. So those 5 time points give you a sort of trajectory of recovery. And then there's 2 coral species, again, Montipora capitata and Porites compressa, these 2 main ones we found variants in. Red is bleached and blue is unbleached. And you can see during bleaching, cyanobacteria get upregulated during that recovery. And so that's an interesting trend for the microbes. And then in some other microbes, when you do a different sort of group of microbes, and you see some of them are upregulated in the healthy corals. And what's tricky about this type of data is you can't assign causality, right? These are just correlative with the condition of the coral. So now we have some really interesting patterns. We know the types of microbes that are upregulated or downregulated in this stress event, and we can actually go forth and ask really targeted hypotheses about what these bacteria might actually do for the coral. And there's all sorts of techniques of getting at this, like sort of like metatranscriptomes. You can actually look at the transcriptomes of the whole holobiont and see if the bacteria are contributing gene expression and physiology to the coral, and that's sort of the next step, is actually figuring out what these microbes do. And so we have a really cool story of patterns, but we can't actually sort of explain bleaching resistance with these patterns. And so now we've moved on to gene expression. So we're really interested in what genes are up and down regulated during the stress event and how that impacts physiology. Gene expression is a pretty good proxy for physiology, and you can measure kind of a lot of cool things. So we took the 3 sort of most extreme time points: May, which is sort of recovered from the first bleaching; October, which is fully bleached again, and then in the next May, fully recovered again. And we did gene expression of these of only the Montipora that hosts clade C. And that's important. If you host a different symbiont, you have different gene expression. And so if we had lumped C and D together in this analysis, we would be getting a lot of differential gene expression just because they hosted different symbionts. And I think you'll see we actually got sort of an outlier, and it's due to that in our analysis. So we specifically targeted Montipora that were predominantly hosting clade C for gene expression. Gene expression is you basically extract RNA out of your samples. You convert that RNA into cDNA. You sequence that cDNA. This is sort of only the genes that are being expressed because you've targeted RNA, and so you know that these things are actively turned on at that time point. And then you can map them back to a genome or a de novo reference. If you don't have a genome, we actually have a genome. And then you can count the number of reads. And so you get all types of scenarios. You get things that aren't turned on, they're not expressed. You get things that are highly expressed, they have a lot of reads. You get things that are just normally expressed, and you get things that aren't expressed at all. And so you could imagine that what we're looking for is in the resistant colonies, what genes are highly expressed, and how is that different from the bleached colonies, right? And that will give us a sense of the physiology that's contributing to coral resistance. And so this work is actually going on right now. We literally just got these figures last week, and so we haven't figured out what genes we have, but we got a ton of data. And so this is really exciting for me, because for genomics, we're working now on the scales of half a terabyte of data, right? And that's amazing in terms of that's half a terabyte of ACs, Ts, and Gs. You have a text file, right, of ACs, Ts, and Gs. That's less than 1K, right? So to get half a terabyte of ACs, Ts, and Gs is an incredible amount of data. And it's not the kind of thing you can look at and be like, oh, here's a pattern. You really got to do a lot of bioinformatics, and it's a ton of computer programming to figure out what's actually going on, what's differentially expressed between these groups, right? And so it's really challenging, both not only from the side of extracting the RNA and making sure it's high quality, but then the analysis pipeline is really tricky, and there's all sorts of caveats and things and parameters you can change. And so we've been messing with all that. We've got some really exciting data. The first thing I wanted to show you is from the coral host. And what you can see is in this PCA is it's kind of a spread, right? And we're not seeing any trends really. The A's are from May, and then the Bs are fully bleached, and then the Cs are fully recovered. And then the numbers are the colony numbers, and again, remember they're pairs, and so 121 goes with 122. Those are right next to each other. We're not seeing anything being driven by the pairs, which I think is really interesting because they're in the same environment, right? They're exposed to the same environment, but we're not seeing clustering based on that. What we are seeing is, if you squint your eyes enough, there's sort of clustering by the individual, and that sort of makes sense. Every individual coral is expressing genes at a different pace, right? And there's a lot of things right now. If we did gene expression in the room, we would all have very different gene expression because somebody, drank milk this morning, and somebody else, was really trashed last night, and they woke up hungover, and that's a different set of gene expression, right? And so you would expect that variance among individuals. This doesn't surprise me. What's amazing is, regardless of this level of variation, we can pick out trends between these pairs. Do the direct comparison between bleached and unbleached colonies, you can measure or differentially express. This first yellow for the bleaching, the mid-stress is during bleaching, the post after. And these are the number of genes that we've are differentially expressed between these two groups. That's a lot. And so during bleaching stress, maximum bleaching stress, there's 1,800 genes that are exclusively found only at that time point between bleached and unbleached. That's a lot. In the literature right now, most people resistance. Missed a lot. Right? And so we're really excited. We don't know if that's because we're using a different species. We did it. Our thing is natural, right? My research is taken from the field. Clearly, there's a lot more gene expression going on with multiple stressors than there would be in a lab experiment where you're only exposing them to thermal stress. But this is real, and that's what's exciting about it. Is these field samples are actually what corals are going through during a bleaching event, and so we're really excited about this data. The host is expressing a lot of genes differentially. Once we dig into this, I'm sure we'll find a fewer number of sort of ways that are impacted because multiple contribute to a pathway. But then we can start sort of looking at gene networks and seeing the once we get to the networks, we can actually tell what aspect of physiology is being impacted during bleaching. What are the corals that resisted bleaching? What are they? Is a really exciting question, right? This is kind of what we all want to know. To corals persist in the future. And at the same time, about 20% of these reads, right, this massive sort of sequencing effort, 20% of these reads come from the zooxanthellae themselves. We specifically targeted colonies that only had C, like I said before. Interestingly, this 71 is a colony that was one of those that shifted. They shuffled from C to D, and you can actually see that in the gene expression. The other 71 here in the beginning is buried, right? And then as it gets more clade D in it, it's getting much more different than the other coral samples. And when it's recovered with clade D, it's very different. So this is really exciting because we have a group of corals that hosted clade D. We didn't spend the money to analyze them quite yet. This is really preliminary data for an NSF grant, and we're hoping to write this into the NSF in terms of we know gene expression is really different when you host a different symbiont because we have data for that in this shuffling colony, right? Exciting about the zooxanthellae is that we got a really distinct pattern in time. And so across all of these colonies, all the A's are the May before bleaching, right? These are very different on PC2 than the zooxanthellae gene expression during bleaching and after bleaching. And so this is pretty cool in terms of we have some real sort of solid numbers here to look at what physiologic responses the symbiont doing during. And then how are those, once we start making gene networks with the symbiont, then we can compare those to the gene networks with the coral and actually see the crosstalk between the two groups of these symbionts, right? And that's really exciting. Right now in the literature, there's only a few papers that have done this with symbionts, and they say there's no differential expression between symbionts exposed to heat stress or not. Clearly that's not what we're finding. We're finding over 6,000 genes that are expressed during bleaching, right? Differentially between those bleaching. So that's a big number. It's a lot more than zero. Pre-stress are many fewer genes being expressed differentially, right? And that's cool. Everybody's doing the same thing. They're all the same clade as C. They're all sort of photosynthesis. They're doing they're tricks, right? But clearly this stress has greatly impacted the physiology of the symbiote at the same time it's impacting the physiology of the coral. So this data is really exciting. This is clearly sort of totally novel. And again, the next stage now is to annotate these genes, figure out what they're doing, do this sort of network analysis, and then start combining that with the corals. It's sort of really, this is, like I say, hot off the press. This figure was made last week. So that's data. I think this is sort of the way forward with all the samples I have. And so, as during this bleaching event, during these two bleaching events, I took the time to collect a bunch of samples and I froze them in liquid nitrogen. Now they're all in the -80, so I have thousands of pieces of coral frozen in -80. I'm sure my boss hates me. And so I can propose this to NSF. We have these three time points. What we can start doing is looking at gene expression during recovery. So we have multiple points during recovery. How does a coral recover from stress? What is it turning on? What is the physiologic mechanisms of that recovery? That's a really important question. We actually have 2 different time points where we can measure recovery. So that's really exciting. Clearly we have differences between healthy and bleached, but that's only part of the story, right? And so this is quite expensive. So this is sort of the basis for an NSF grant we're working on right now. So these genomic tools give us this really great sort of insight into the mechanisms of resistance, right? And this resistance is a critical component of resilience, right? And like I say, these tools could be applied to any creature. Now gene expression is being done with all sorts of things, plants and animals. It's not trivial to extract RNA out of a creature, but it's certainly not impossible. I figured it out, so I'm sure any of you guys could do it. So that's exciting, but again, this is just one piece of the resilience picture, right? What we also want to know is how do coral communities, how do corals recover from stress? And a lot of the work I did with Valerie Paul and before my PhD was to look at this mechanism of recovery because I'm really interested in larval ecology. And so corals are kind of a crazy creature to study for larval ecology because many of them spawn their gametes, and so they release eggs and sperm into the seawater. These gametes fertilize in the water. They develop into embryos and planula larvae. These larvae float in the plankton for 2 weeks. This is a pretty special event. Coral spawning only happens sort of once or twice a year. It's tricky. It happens at night, a few nights after the full moon. It's always kind of variable, so you got to be in the water a lot at night, which is a little freaky, but totally doable. And so I've done a lot of this kind of work to collect the eggs and sperm, and then do a bunch of experiments. We wrote this review a few years ago now. Wow, it's 10 years ago now. Wow, it doesn't feel like that. And critically, what we wanted to point out with this review is that there's multiple life history stages that have to survive and persist for become part of a new community, right? So it's not only just that they need to be sort of spawned, so fecundity is important, fertilization success is important, settlement is important, where they like to settle, that habitat, and then growth and survival. Into all of these things are key factors into coral recruitment. So if we expect recovery on reefs, we have to manage all 3 of these things. We can't just ignore one and plan for the other. And so a lot of my chemical ecology work has been focusing on this sort of settlement ecology process. And I'm really interested in crustose coral and algae. These are sort of often ignored benthic creatures that live on reefs. They're encrusting red algae. They're very hard to identify, and I think that's why they're being ignored. People are lumping them together. They're saying we have crustose coral and algae on this reef. That's great. There's over 30 species in the Caribbean. So clearly, when you say there's crustose coral and algae, I would ask you, well, which species is that? And they're like, we don't know. It's the pink one. I'm like, well, that's not very helpful. We use these teeny little mounds. These are called conceptacles. It's their reproductive organ. We use those to identify them clearly in the field. This is a picture under a microscope, and you can barely see the conceptacle. This is really hard to see in the field, almost impossible, right? And that's a real challenge to understanding the dynamics of crustose coral and algae communities. Something I'm really interested in is getting at community change in the face of climate change, because these things are calcifying organisms also, and they are heavily impacted by ocean acidification, and we know that in literature. We've never looked at different species. We've only ever used one model species. How are different species impacted by ocean acidification? We have no idea, but that's a really interesting question, right? The reason that I think they're so important is because they're mediating this transition from a coral planula. This is a planktonic stage. It floats around in the plankton. It gets dispersed, and then the planula sticks its head on the bottom, and it metamorphoses into a single polyp. This is the beginning of a coral colony. Single polyp settles, metamorphoses, and then grows and grows and grows. These, what we think is going on is these planula are little sensory units, and they make the decision within weeks to months of where a coral is going to live for the rest of its life, up to thousands of years. So clearly there's been a huge selective pressure for them to choose wisely, right? If you settled in a poor place and you died in a day, well, you're not doing too well. You're not going to survive, right? But if you're going to live for thousands of years, you need to find the right habitat. What I think is going on is certain species of coral and algae actually require the same things that corals do. They require the same water flow. They require similar light environments. They want low sedimentation, right? And so if you find a coral and algae living in a place, that's probably a good indicator species. I call it sort of neighborhood effect. When you guys go out and you shop for a house, you look around and see who your neighbors are, and you're not going to buy a house if you don't like your neighborhood, right? And that's kind of what the corals are doing. They're using these coral and algae as an indicator species. And so we did this experiment with a bunch of different coral species and a bunch of different algae to see if this trend is true across sort of all the species. So in this case, I've normalized settlement on the coral and algae, and I've divided it by settlement on a biofilm, because I'm assuming a biofilm is the de facto presence on benthic ecology in coral reefs. Everything has a biofilm on it. And so I've sort of normalized among my experiments. Each of these is a different coral species from the Caribbean. The yellow bars are those that spawn their gametes, then fertilize in the seawater, and then the blue bars are brooding larvae that have internal fertilization, and they settle much more rapidly. The dotted line is at 1, and 1 would mean that there's the same settlement on coral and algae as there is on a biofilm. So basically, these larvae will settle on just about anything, right? If the bar is higher than 1 and it has an asterisk, it means it was significantly different than the biofilm. And if the bar is higher than 1, it's an inducer, right? So that's facilitating settlement. If it's lower than 1, it's an inhibitor. And so for this species, Hybulithon borgessenii, it's clearly facilitating species of corals, right? All the spawning species of corals are responding that way. All of the brooding species settle on it too. They don't mind it. It's not hurting them, but maybe it's not quite as important in their settlement ecology to choose that space. We see this true with Titanoderma prototypum, another species of coral and algae that we see in the field, but both of these species are really quite rare in the community. So when you take the most common species, Perigoniolithon solubili, then we start seeing the opposite. It's actually inhibiting settlement for some of the coral species. Even though this bar is really large, it's actually 2 very small numbers, and so it happens to be that the denominator is much smaller than the numerator, or whichever way that goes. So it looks big, but it's actually not significant at all, right? And so in this case, the coral and algae is actually worse for these 2 species than just a biofilm thing. So not all coral and algae are settlement facilitators and inhibitors. Species matter, right? And that should be a no-brainer, but it's not in the literature. And so a bunch of my papers have sort of shown that. What controls the abundance of these facilitating species? I put this slide up there because I have no idea. I'm hoping somebody someday in the audience will come up to me and tell me the answer. I know that's not going to happen. So this is a great these are great student projects, right? These kind of questions: how do coral and algae compete with other things? How do they compete with macroalgae? What do they require to live? Are those conditions true now on coral reefs? Do we have conditions for these facilitating species? Have we already shifted away from a natural habitat that's driving these facilitating species away from being present on natural reefs? And is that what's driving the loss in recovery of coral reefs, right? These are great, excellent questions for students, and clearly going to be a future part of my lab. We've also bothered to test macroalgae to see what its impact is on coral recruitment. And so when you do reef surveys, you get some sort of relative abundance. In this case, we had about At this reef in Belize, we had about 18% coral cover and some gorgonians and a pretty small amount of coral and algae, less than 10%, but clearly a huge amount of macroalgae. So if you want to know coral recruitment, you've got to test this thing because it's the dominant creature on coral reefs, right? And so we can do these cool sort of experiments in situ. We can put a bunch of coral larvae in this acrylic tube. We've put Nitex mesh on each side so that these tubes are getting a little bit of water flow. They're exposed to natural sunlight, and they're exposed to natural temperature in the field. And then we can put a settlement substrate. We know they like this hydrolithon species of coral and algae. And then we can add an algae to it and see, does that algae deter settlement on something they like? And we can do these kind of experiments over and over again with different algae species, different coral species. We've published quite a bit on this. We found that not only are the algae inhibiting recruitment, but the natural compounds found in these algae, the natural products, are also inhibiting recruitment, and some of these are toxic. So we did a ton of this work with cyanobacteria. I collaborated a lot with Valerie Paul, and she's really interested in the natural products from cyanobacteria. So we had sort of in our lab to isolate novel compounds. And this is really exciting. These compounds are really hard to isolate. They're hard to structurally characterize, but we had a great lab full of sort of multiple experts. And so I could go out and test these individual compounds against coral larvae and see if it's actually the single compound itself driving this interaction. Because macroalgae has a lot of properties that might be bad for coral larvae, right? We don't know what those are, so we wanted to nail down what is driving this interaction. So we can actually isolate a compound. We can test the larvae. Again, this is normalized, and so this is the amount of settlement in the presence of kerosene D divided by the control, which is just a sort of a biofilm conditioned tile. And one would be if there was just as much settlement on the control as in the presence of this single isolated compound. We embedded these compounds in agar strips, and we attached them to the tiles because these are all nonpolar. And you can see clearly nothing was as good as just the conditioned tiles, but not all of these are significant. These three compounds are highly significant in terms of they are actively killing or deterring coral larvae from settling on our reefs. So you can imagine now we're not only understanding that macroalgae are bad for coral settlement, macroalgae and cyanobacteria, I should say, but also the compounds they make are really bad. And so this is the mechanism of how, right? How do these macroalgae inhibit coral recruitment? We can do a lot of interesting thermal stress experiments with coral larvae and see if thermal stress is bad for the amount of coral settlement. And what was kind of interesting about this series of experiments, we did multiple experiments like this in the Florida Keys, was that we didn't see an effect of survival associated with a +3 degree treatment. And that was kind of interesting, and I was a little surprised, frankly, because in the literature there's a lot of sort of thermal stress is bad for coral larvae. We published this already in multiple papers. What's important is that the thermal stress is actually creating a sublethal stress. And you can measure that through antioxidant enzymes, upregulation of antioxidant enzymes, and endpoints like lipid peroxidation and lipids, right, critical for cellular membranes, proteins, right, critical for cellular function. We've seen upregulation of these sort of endpoints. So even though temperature doesn't kill the larvae, it's hurting them. And that sublethal stress could be really important in the context of climate change where multiple stressors are interacting to affect larvae. So we took this compound microcolon A. We knew it was bad for the larvae, and we combined it with a temperature stress. So we were really interested in what happens when you have multiple stressors on the reef, competition and climate change. Is that worse than just competition alone? Is that worse than just climate change alone? We get a figure like this. The first bar is the survival of the coral larvae in our ambient conditions, 27 degrees and no microcolon A. The second bar, all the blue bars are added temperature +3 degrees. You can see, again, we get the same trend. There's no mortality associated with temperature. We sort of knew that from previous experiments, but we know that there's some sublethal stress, and in this paper there's a bunch of antioxidant enzymes we measured. You add in microcolon A, the single compound, it impacts the coral larvae quite a bit. It's toxic to the coral larvae. You do them both in combination, temperature and microcolon A, and you get a synergistic interaction. And so we can determine this through our two-way ANOVA, right? And we see that this synergy is actually killing way more larvae than either stressor alone. And that's really scary, right? When you look at the settlement data, it actually drove the settlement, this interaction term drove the settlement to below 5%. So you can imagine if 100 larvae are released on the reef and less than 5 of them settle, and that's just settlement, right? We're not even looking at post-settlement survival and growth. If we're bottlenecking our populations down to less than 5%, it's going to be really hard for coral reefs to recover from climate change. So clearly habitat, both positive and negative cues, are really critical for this component of resilience, for coral recovery from sort of change. And so I just want to touch on this. It's sort of a hot topic now in coral reefs is should we be using restoration to actively sort of manage coral reefs? I would argue yes. Frankly, we sort of created this problem, and so maybe we should be sort of helping to solve this problem. There's a lot of argument about how to do restoration, and I don't know that I know the best way. I think at least these 3 measures have been thrown out there. We can sort of drive resilience through restoration. We can add in sort of more corals. We could actually target certain corals that we know are thermally tolerant and move them to another place and increase that thermal tolerance by moving those alleles over there. And then we can actually selectively breed. This was sort of a concept that my advisor suggested. We could selectively breed corals to increase their resilience. I think there's some moral issues associated with all of these, and just like anything that humans have tried to intervene in, clearly we've made a lot of mistakes, right? So I wouldn't necessarily recommend any of these things, but these are all really terrific pure science type questions that have an applied sort of basis. And so these are kind of exciting. I've been working with this group Seacor. We go to places like Curacao, Bahamas, and Mexico, and we do coral spawning. We collect the larvae. We settle them on these substrates that we can plant on the reef. And then what's really nice about this restoration technique is we're ensuring genetic diversity. What I worry about with most coral restorations is that they're planting fragments, and that means they're taking the same genotype, making 100 replicates of that genotype, and planting 100 of that genotype on the reef. If that genotype is susceptible to disease, you haven't actually solved the problem, you've just delayed the problem for a few years. The advantage of using sexual recruits is that you're incorporating genetic recombination into every generation, and so you're increasing the genetic diversity on the reefs, and I think that's critical for adaptation. We know through population genetics that's critical for adaptation. And so this is a really important thing. Clearly here in Alabama, you guys care about oysters. I care about oysters too. I don't know that much about them, but they're calcifying organisms. They have these same types of questions. I just threw that in there to highlight that all of these techniques that I showed you in this presentation can be applied to oysters. Any creature, really. Seagrass, you can use this with seagrass. You can do sort of any type of creature you want. And I think what's critical that we need to keep in the back of our heads as sort of restoration practitioners is how do we maintain this genotypic richness that's found in natural populations, and how, instead of just continuing to whittle down to certain genotypes, we need to actively be trying to increase the genetic diversity, because as more and more threats are threatening the marine environment, there's going to be more different types of interactions and different things going on, and we can't predict all of those things. But fundamentally, we know through population genetics that genotypic richness is really critical for adaptation and persistence. And so this is kind of the thing to keep in the back of your head during restoration. Clearly, millions of people helped me, and they're all awesome. Thank you to all of them, and I'd be happy to take any questions.